Hello, well, let's get started now. Good afternoon, everyone. My name is John Voss, and I'm in the Department of Biochemistry and Molecular Medicine in the School of Medicine. And it's really my honor, and I'm just, I feel very, very grateful that Ron Kaback has accepted our invitation to be a Storer Lecturer here, which is our premier life science forum for really distinguished scientists and researchers in the life sciences. I happen to be, just a little background, I happen to be a postdoctoral fellow in Ron's lab from 1994 to 98 at UCLA, and obviously a great impact on my career. So I'm very, again, very grateful that he's here, and I know he's had a nice time today meeting some of the people here at Davis. A little background. Dr. Kaback received his BA from Haverford College and an MD from Albert Einstein School of Medicine. He was a postdoctoral fellow at Einstein and then a commissioned officer in the United States Public Health Service. So I never got to see you in your Navy whites. Yeah. Yeah, that time of history, I guess. But that got you affiliated with NIH, so you started at NIH and eventually became a senior research investigator there. And then in 1970, he joined the Roche Institute of Molecular Biology, which was the only big pharma research institute that was devoted purely to basic science, basic research. So I guess you could say non-translational in one regard. But he was there, and he ultimately became the head of the biochemistry department and the director of a laboratory of membrane biochemistry. And then in 1989, he finally came out west to become a Howard Hughes Medical Institute investigator at UCLA, where he currently is now as a professor of physiology in the School of Medicine. So Dr. Kaback is world-renowned for elucidating the molecular structure and mechanism of ion-coupled membrane transport. Along the way, he has made many important contributions to membrane biochemistry and biophysics, such as Kabackosomes, membrane vesicles, which are membrane vesicles that not only provide a manageable system to study membrane transport, but also have proven invaluable to the field of bioenergetics as well. He is greatly appreciated for the development of probes and platforms to measure electrochemical ion gradients in microscopic systems, and most impressively for his application of emerging techniques to understanding the mechanism of action of membrane transport proteins. And from my perspective, I guess that would be one of the most important contributions in the fact that Ron was always, and always continues to this day, to be willing to look at any new approach that will give him information on his beloved lac permease. Dr. Kaback has been the recipient of many honors, including the Selman A. Waxman Honorary Lectureship Award by the American Society of Microbiology, the Louis Rosenthal Award by Brandeis University, the Kenneth Cole Award by the American Biophysical Society, etc., etc. Notably, he was elected to the National Academy of Sciences in 1987. So, without further ado, I'd like to welcome Ron to give us a little bit in the first of his talks, which will be hopefully very historical and appreciated by everyone here.
Well, John, thank you very much for a very nice introduction and for really a very nice invitation to be here. It's my pleasure to be here, at least as much as it's your pleasure to have me here. And I'll try to entertain you for the next, I guess, an hour or so, and try not to put you all to sleep, okay? I'll do my best. And if I notice it's a poor Goldcorn falling asleep in the first row, I will know it's time to quit, okay? So I'm going to, the idea of this title was in case anybody would think that I was going to tell them how to charge their cell phones. that they might show up for this lecture, okay? And I will try to be as humorous as possible. But let us start with the experimental system that I'm going to talk to you about, which John already told you a little bit about. So membrane vesicles, I have to tell you about membrane vesicles, and I'll try and do this. I'm going to go quickly at first, and then I can't stand being behind here. I will continue and try to go slower when things get more difficult to understand. So here's an idealized E. coli showing its surface membranes. What we're after is the plasma membrane, that membrane that surrounds the cytoplasm of the cell. And what it really looks like, here's a real E. coli. And as you can see, what we're after here is this membrane, what's in red. And there's an outer membrane in gram-negative bacteria, as many of you may know. This we want to get rid of and end up with this membrane. So how do you do that? You do this by osmotic lysis. So to get that red membrane, you make a spheroplast with either lysozyme EDTA or penicillin, and then lyse the cell osmotically in a very controlled fashion, and you end up with these membrane vesicles called by some Kabackosomes. I never use the term primarily out of modesty, okay? However, these Kabackosomes transport all kinds of stuff. They transport lots of things. I'm going to end up telling you mostly about lac permease and lactose transport, but they transport all kinds of amino acids. They transport lots of stuff. So the way this works actually is that if you take spheroplasts or cells and mix them up with colloidal gold, which you can see in an electron microscope, the gold is all outside because, of course, the membrane is very impermeant to something that big. But if you lyse the spheroplast in the presence of colloidal gold, you end up with colloidal gold inside of the vesicle. So there's a big rip in the membrane. The guts come out, and then it reseals, and you form basically what's a ghost of E. coli. And these things have very little, if any, cytoplasmic protein. They will transport when you get to the right conditions, just like whole cells. And first, let me tell you that they're all right side out and give you some characteristics. So these are freeze fracture electron micrographs, which were taken probably before a lot of you were born, 1966, right down there. And the texture of the convex surface always looks this way, and the concave surface this way. So this is what the membrane looks like in intact E. coli. So this was the first evidence that they were really all right side around. And this was a big controversial point at one time in my career. Now, one of the things that came out of this was a freeze fracture in 1971 that showed the active state of the membrane, okay? And it says up here, it sort of indicates that the membranes may be Jewish, okay? For those of you that don't recognize this, it's supposed to be funny, okay? It's on purpose this way, okay? So now you can't really say that the membranes are Jewish, okay? Because a lot of different religious sects use circumcision, okay? However, a few years after showing this at every talk I ever gave, this was presented to me by Joel Cohen at the University of Pacific, and clearly this is from endoplasmic reticulum that he took this electron micrograph of this six-pointed star, which is probably a virus particle, but it does prove that membranes are Jewish, okay? An important point in my talk, okay? So these vesicles are all right side out, as in an intact cell, as demonstrated, as I already told you, by freeze fracture electron microscopy. We did an exhaustive study with 22 different antibodies to specific membrane proteins, and you can show that the ones that are supposed to be on the cytoplasmic side, there can't be any more than 2% of the membranes that are either leaky enough to let antibodies in or broken, okay? They're really right side out, and to really nail that, each membrane vesicle catalyzes active transport. This is something that took about 20 years to figure out how to do, and it is a really wonderful experiment, which I'm going to show you in a second. First, I have to tell you that there are at least 4 different kinds of bacterial transport systems that transport small molecules. The first one that made vesicles really fashionable was the phosphoenol pyruvate phosphotransferase system, which is a vectorial phosphorylating mechanism that exists only in bacteria. Okay, and it's important not only for transport and the first step in metabolism of glucose and other sugars like it, but is also very important for catabolite repression. I'm sorry, I'm standing where you may not be able to see. Okay, so the second type are these ABC transporters, which I'm sure all of you know about. They utilize ATP in a multi-subunit components to drive one-way transport of certain sugars and amino acids through the membrane. And then there are ion-coupled ones, the ones we're going to talk about, which are the sodium-coupled glucose transporter, which is a eukaryotic protein, but also found in bacteria. And then finally, there are proton-coupled systems like this one, which we're going to talk about in some detail, mainly tomorrow. But I'm going to use this in a moment to tell you lots of things. So one of the ways that you assay transport, just so you get a feel for what we're going to talk about, and this is really sort of important, the assays that you use, as you'll see, is you have usually a radioactive substrate, which you have 50 or 100 microliters of membrane vesicles. You add your radioactive substrate and some kind of an energy source, which we're going to talk about in detail. And at a given time, you dilute and you filter this through millipore filters under vacuum, take the filter off, and you count radioactivity. So that's the usual kind of assay that's used. Okay, here's what a typical transport experiment looks like. And this is actually an original one that was done a long time ago. So what the big trick with this was to discover that in order to get these things to work really well, you need to use D-lactate oxidation. Now, no one in their right mind would use different isotopes of, different isomers of lactate or anything else. At the time, we used, like everything on the shelf I used, to see if I could get transport to work, and I don't have time to tell you how this was discovered. But let me tell you that D-lactate oxidation specifically will drive transport like crazy. This is a filtration assay. If you don't add anything, this is lactose uptake in membranes that have lac permease, which you're going to hear all about tomorrow. You get very little transport. You add D-lactate and you get this huge stimulation of transport. This represents a 100-fold concentration gradient in these empty vesicles, and it's all free lactose, because if you add cold lactose and you're looking at radioactivity, the radioactivity all comes out. Or certain energy poisons like dinitrophenol, which are protonophores that you're going to hear about in much more detail, everything will come out. The D-lactate is converted almost stoichiometrically to lactate. And in bacteria, like in mitochondria, the respiratory chain is in the membrane. So what's happening here is you have a single-step dehydrogenation, feeding electrons into a membrane-bound respiratory chain, and magic happens. You get a great deal of transport. And at this point in my life, things were really in a state of controversy in the bioenergetics field. There were maybe a half a dozen biochemists who had all the power in biochemistry, who believed devoutly that there had to be a high-energy intermediate involved in oxidative phosphorylation, which this clearly was related to somehow, maybe because it's respiratory-driven work, okay? And there was a guy named Peter Mitchell, okay, who came up with this crazy hypothesis called chemiosmosis, which I'm going to tell you about in some detail. But at this time in my career, I didn't understand what Peter Mitchell was saying. And I had a lot of evidence against chemiosmosis, okay? And I had this hypothesis that the lactose permease in particular was a respiratory intermediate. And this became a big theory that was actually in Leninger's 1970 textbook, okay? That's how famous it was. Well, what happened was, first of all, I have to show you that things were sort of casual these days. This is actually me in 1975. We had a Polaroid camera at the time, and we were screwing around with it, as you're going to see with other photos. This is when I had a red afro and a red beard, and I wasn't limping or using a cane either. So anyhow, on to the next slide. So You can drive transport with other things besides D-lactate. So artificial electron donors like ascorbate PMS are even better than D-lactate because there's uncontrolled feeding of electrons into the respiratory chain. You can use ubiquinol coupled systems. You can even get anaerobic transport to work. You can get anaerobic transport to work. If you generate ATP internally, which I'm not going to tell you about, you can get it F1FO to make a proton gradient and drive transport, which I'm getting ahead of myself. And you can make artificial gradients, which at that time were really important. Okay. Now, I wanted to tell you how to show that each vesicle catalyzes active transport. And this comes from a series of experiments, which I'm not going to tell you how they began. But if you take a compound that looks like this, it's called vinyl glycolate. So it's an analog of lactate, but it has a double bond between the third and fourth carbon. There's a lactate transport system in the vesicles, and you'll get transport as the unchanged compound. But inside, you have these lactic dehydrogenases, which will convert this into the analog of pyruvate, which in this case has a conjugated unsaturated system and is very reactive and can be attacked by nucleophiles at that carbon and will label all kinds of things, okay? So what we did at the time was to make radioactive vinyl glycolate, this stuff. And you can show that the rate limiting step for covalently labeling the membranes is transport, okay? This is very important. So if you don't add anything, you get this sort of lousy uptake. If you add, in this case, ascorbate PMS, you get big labeling, only there's a big difference between this and what I showed you before. And that is that if you take the membranes at this point and you boil them or you add cold stuff, nothing happens. It stays there because it's all covalently labeled. If you add dinitrophenol or ascorbate PMS without doing anything, it goes back down to here. So what was done was to take this stuff, make it very radioactive, and then do radioautography in the electron microscope with vesicles that were incubated under these conditions here. And what you get is this image of all these black worms which represent radioactive decays of tritium. And obviously they're localized over every single vesicle. If you look at all, lots of fields, you see this huge concentration of worms, black stuff over the vesicles, indicating that every single one of them transports, okay? So this is a unique kind of situation which took like 20 years to figure out how to do, because everything else comes out when you try to fix the things. Okay, so powering the membrane. Now we're going to talk about stuff that's going to get a bit hairy, because I'm going to explain less stuff to you. You will all understand what I'm saying as I'm saying it. But 10 minutes after you leave this room, you will not be able to repeat any of it. So it's like, I tell you that ahead of time. But you're going to get a feel for what chemiosmosis is all about. So this guy, Peter Mitchell, whose picture is shown here, came up with this crazy hypothesis, okay? And his hypothesis, which is shown here in brief, is that respiration or light, the two basic energy trapping devices of living cells, gives rise to an electrochemical proton gradient, the equation for which is shown in here. And if any of you are afraid of equations, don't be, because this is really damn simple. Okay, it's exactly what it says it is. It's the delta mu H means electrochemical gradient, which consists of both an electrical component, a membrane potential, delta psi, and a chemical component, a pH gradient. And all the rest of this garbage is constants that puts everything into millivolts, which I like to put them into. You can put it into joules, you can put it into all kinds of stuff. I don't understand those things. Millivolts I understand. This delta mu H is supposed to be, according to Peter Mitchell, was the driving force for oxidative phosphorylation, which it most certainly is these days. Active transport, which you're going to hear all about until you get sick, transhydrogenase activity, the bacterial flagellar motor, the efflux and influx of proteins through a membrane, the efflux of antibiotics, all kinds of stuff. This is, in fact, the other paradigm. You all know about DNA, RNA, protein, which, of course, is one of the two paradigms in biology. The other one is chemiosmosis, okay? But you get short-shrifted on chemiosmosis because it ain't easy to understand. And you can't crystallize an electrochemical proton gradient. All you can do is measure it, okay? So it's not something biochemists would like. I didn't, for sure. So the big question was, in 1970 or 1965 or whenever it was, is how do you measure membrane potentials and pH gradients in microscopic systems? This was really the big problem at the time. And that's what I'm going to tell you about. I'm going to give you a feel for how you do this. So you measure membrane potentials by using so-called lipophilic ions, which were invented by Russians, Lectureship and Lieberman in Moscow. And these are compounds that are like tetraphenylphosphonium or tetraphenylarsonium or the methyl triphenyl derivatives if you want to measure a membrane potential that's interior negative. These things can delocalize charge so that they can get through a membrane. And what you do is you make them radioactive, okay? And then you look at transport, just like I showed you. And if it's negative inside, something like tetraphenylphosphonium will accumulate in equilibrium with the membrane potential, and you can measure, you know what the internal volume is, so you can measure the concentration, you can convert that into millivolts, and you've got a number, right? And you're going to see these things later. If the membrane potential is inside out, as it is in chloroplasts, which some of you may well be interested in the audience, it's internal positive because the protons get pumped in, and you use tetraphenyl boron or thiocyanate, and it's the same business, okay? These are permeant ions. You can demonstrate that they're permeant with liposomes. You can do all kinds of things with these, and you can measure membrane potentials. You can do it actually by filtration, because when you do filtration with a charged compound, it's difficult for them to leak out when you do the dilution step. And this becomes very important later, because here you want to measure now pH gradients, and the way you do this is simple, okay? If it's supposed to be internal alkaline out, so like it's shown here, so acidic outside relative to inside, you use a permeant weak acid like acetate. So acetate on the outside is obviously has an equation where it's protonated or unprotonated, and there's an equilibrium. It's the protonated form of the acid, which is permeant. It's not charged. They can penetrate the membrane passively. If the inside is alkaline relative to the outside, you cause dissociation of the acid, and the anion can't get out, and that's radioactive. So you measure uptake. The opposite, you use a base if it's interior acid, okay? It gets protonated inside. The protonated form can't get out. You can measure that. For 7 years, I had every postdoc in my lab try to measure pH gradients in membrane vesicles, and they were uniformly negative. And this is because every time you dilute to filter, if you're going to use filters, all the acids leak out. They're very permeant. They leak like crazy. So what happened was that Gary Rudnick, who was a postdoc in the lab, introduced a technique that was developed by Kolowick and Womack in 1969 called flow dialysis, okay? We were measuring binding of galactosides to lac permease with this, and it's a very simple, really simple technique. And I want you to understand it. Some people, for some reason or other, refuse to understand this technique. It's simple dialysis, except that you flow buffer through the bottom chamber. This goes to a fraction collector, which you count radioactivity. So what happened was we were doing binding, and I got the bright idea one Friday afternoon that this technique is perfect for looking at steady-state concentration gradients in membrane vesicles, particularly of weak acids, because you don't screw it up. You can't dilute it. All you're doing is injecting radioactivity into the top where you have your membrane vesicles. The membrane vesicles can't get through the dialysis membrane, but the weak acid can. And you're constantly measuring the concentration of the weak acid bathing the vesicles because you're measuring what's coming through the dialysis membrane continuously. Now what this looks like is really beautiful, and you'll see that in a minute, but here's a picture of Rudnick when he was a hippie. He's now a professor at Yale and doesn't look this way anymore, okay? But this is how he looked in my lab, okay? I can't resist these pictures. So one of the things, before I get to all of this business about chemiosmosis and how you make these measurements, I want you to know a little bit about ionophores, which turn out to be very valuable. So one of them that we use and that you'll see is valinomycin, which is a cyclic dipsipeptide that's very specific for potassium. It makes membranes permeable to potassium, okay? Nigericin is an electrically neutral ionophore that exchanges mostly potassium for protons, but it'll also work with sodium. And monensin is the similar kind of ionophore, but its preference is for sodium instead of potassium. This one you won't see again. That's just up here for students. Okay? So now, what does flow dialysis look like? Here's what it looks like, and it's damn simple. Okay? So you have, on the top is supposed to be pictures of what a big giant vesicle would look like in the upper chamber, not able to get through, and the yellow represents, in this case, acetate. And this is an experiment that was done after one Friday afternoon. I got the idea that this technique would be perfect for looking at steady state concentration gradients, particularly of weak acids like acetate. And at that time, Shimon Schuldiner, whose picture you'll see soon, was a postdoc in my lab, and every time I would suggest an idea to Shimon, he would say, nah, it'll never work. Won't work. So this was Friday afternoon, and Saturday morning I got a phone call from Shimon saying I had to come into the lab because there's something really amazing. This is what was amazing. So what happens is you have your vesicles, right? And you do a run with or without vesicles, and you get the purple line, okay? The purple line runs like this, which means that that this is with acetate being injected in at 0 time. So it goes into the lower chamber, reaches a steady state, and dialyzes out in semi-logarithmic fashion. It looks linear, but it's not. Okay? So what's happening is that there's no binding of the weak acid to the membrane vesicles, because if that were the case, the yellow line would be lower than the purple line, right? Because this is where we have membrane vesicles, the purple's where we don't, and they're the same thing. If there was binding, the dialyzable concentration would drop, and you'd see less. You add ascorbate PMS, okay, and there is a huge drop in the dialyzable concentration of acetate. It reaches a new steady state, and it will run parallel to this line until you run out of the ascorbate PMS, or in this case, we add valinomycin. I'm showing you this just so you'll believe what I'm going to tell you later. So valinomycin allows potassium to go in, which collapses the membrane potential. And what happens when you do that is you increase acetate uptake, indicating that you increase the pH gradient, okay? So these are reciprocally related, in other words, all right? You then add nigericin, and everything collapses, and in a lot of runs it'll overshoot like this and then come back down. This one didn't happen to do it. Okay, so this is how all these measurements that I'm going to tell you about, with tetraphenylphosphonium, with acetate, with different transport substrates, they're all done this way. And it's not good for kinetics, because you're measuring, this is all thermodynamics, right? You need to know how big the membrane potential is, how big the pH gradient is, and how big the concentration gradient of a substrate is, okay? That's what you need to know to really test this hypothesis. And here is Sophie. We had a good time with the Polaroid camera, as you can see. This is Sophia Ramos, who did a lot of these experiments, and Shimon Schuldiner, who at this time had hair on his head. He's now bald. Not quite as bald as John, but bald, okay? All right, so one of the things that was really impressive to me is this business about you add valinomycin and you get an increase in the pH gradient. Now in mitochondria, there's something called respiratory control, which means that the magnitude of the electrochemical proton gradient is in equilibrium with the rate of respiration, the rate at the proton pump goes, right? So if you have a big delta mu H, you have a low rate of respiration, and if you add an ionophore, the rate of respiration goes up, and something else happens, like an increase in delta pH, which is what's occurring here. But, however, in in E. coli and in E. coli membrane vesicles in particular, there's no respiratory control. Respiration just keeps going. So you look at this thing for 5 years and you figure, Jesus, what in the hell is going on? What's happening? So here's what's happening, and it's so simple that it's amazing that it took me so long to understand how it works. So here's an idealized membrane vesicle, obviously, and on one side there's a membrane potential, and on the other side I made a pH gradient that's alkaline inside. Now I put it to you that each one of these forces is a force that limits the magnitude of the other. In other words, an interior negative membrane potential will limit the magnitude of the pH gradient because it's going to want to pull positive protons back in. By the same token, when you have a big pH gradient, there's a diffusion pressure of positive charge going in that's limiting the magnitude of the membrane potential. So if you cut out one, the other one goes up automatically. And you can actually demonstrate this in liposomes, okay? You don't need proteins at all for this. It's very straightforward. And I show it to you because I want you to believe what I'm going to tell you. So first, before I tell you all kinds of things to confuse you, let me just show you that it costs you something, energy. It costs you energy in the form of the proton electrochemical gradient to get transport. So here's a flow dialysis run. Again, this is with acetate. You can do the same thing with tetraphenylphosphonium. Of course, we did everything in both ways, but this is, you do the inverse of what I'm going to tell you. So you start by adding acetate at the beginning, only in this case we add valinomycin also from the beginning. So all you have now is a big pH gradient. No membrane potential, because you have to, in order to do what I'm going to show you, you have to clamp the electrochemical proton gradient in one form or the other, okay? So here, with membrane vesicles, instead of adding valinomycin, which was added at the beginning, like I showed you before, we added lactose to membrane vesicles that have the lac permease. And you get a partial collapse of the pH gradient. If it's TPP, you get a partial collapse of the membrane potential, but you add nigericin first, okay? And this will run parallel to the control line, but you add nigericin and let it go the rest of the way. Now this is all due to transport of lactose, because if you take membrane vesicles that don't have lac permease, nothing happens when you add lactose. Same thing over here with glucose 6-phosphate, another substance that's transported by the vesicles. If you have membranes that have this transporter, you get partial collapse of the pH gradient or the membrane potential, and if you don't have the transporter, nothing happens. So it costs you something visually right here in terms of the proton electrochemical gradient to get transport to work, right? Okay. Now, one of the really interesting things about this is that, you know, E. coli is a neutrophile. There turns out to be 3 classes of bacteria: acidophiles, neutrophiles, and alkaphiles. E. coli grows between pH 5 and pH 8 approximately, or 8.5, okay? So what was done at this time was to look at the effect of external pH on the components of the proton electrochemical gradient. And this was a really, really amazing phenomenon, actually. So let's look at, these are all flow, each one of these points is a flow dialysis experiment where you're measuring steady state concentration gradients. And what happens to delta pH is amazing, okay? Because when you think about it, it's not really amazing, but it was amazing at this time. Delta pH is high at acid pH, at pH 5, 5.5, and then it drops precipitously, and at pH 7, 6 actually, it's nothing. There's no delta pH, right? Now this is sort of, it makes sense actually, because you want the inside of the cell to maintain a constant pH. A lot of enzymes don't work at pH 5, okay? So at pH 5 you have a big delta pH because you want the inside to stay at neutral, and that's what it does. It stays at pH 7, 6, right there, okay? So what happens as the delta pH drops, the membrane potential goes up. In this case, this is the only difference between vesicles and whole cells. In whole cells, the membrane potential goes up actually enough to compensate completely for the drop in the delta pH. In membrane vesicles, probably due to conductance of the membrane or something, I don't know really, we don't really know what. But I should tell you, I forgot to tell you incidentally, that all of these measurements were confirmed by other techniques. So the measurement of the membrane potential was confirmed by actually sticking electrodes into giant E. coli with Cliff Slayman at Yale a number of years ago. And we got within 10 millivolts of what we got with TPP. And the pH gradients were confirmed by experiments from Schulman's lab when he was still at Bell Labs by phosphate NMR. Okay? So they all came out the same. So all these measurements are really confirmed by two techniques at least. Okay? As a result of these changes in the pH gradient and the membrane potential, delta mu H in membrane vesicles is maximal at about 220 millivolts, okay? And it goes down to about 120 millivolts at pH 7.5 or 7.6, okay? And I would like you to remember a couple of things. One, we're going to take membrane vesicles and put them at pH 5.5 or pH 7.5 or 7.6, okay, and see what happens with these ionophores that I've been telling you about. And this is where stuff gets hairy. Let me warn you ahead of time. You get this is called the data machine, because I'm going to show you how you correlate all of these measurements with real transport, okay? And that's what we're going to see. So what happens in these experiments is we take membranes at pH 5.5 or 7.5, and they're in potassium phosphate. That's it, okay? Either at pH 5.5 or 7.5, and you start adding valinomycin. And again, as I've told you before, these are in potassium, you add valinomycin, you short circuit the membrane potential because the potassium can go in and out as fast as the protons are being pumped out. So at pH 5.5 or 7.5, the membrane potential tetraphenylphosphonium uptake drops like crazy, okay? As this happens in many membrane vesicle preparations, the pH gradient goes up at pH 5.5 because of what I showed you before. It's a reciprocity business, okay? As a result, delta mu H hardly changes, very small. Next panel, you add nigericin. At pH 5.5, the pH gradient drops and the membrane potential goes up. At 7.5, nothing happens because there's no delta pH to convert into delta psi. And at pH 5.5, you get this, again, a slow inhibition. The last one is CCCP, a protonophore, which works on both. Collapses both of them, works better at 5.5 than 7.5 because of the PK of CCCP, okay? All right, so Mitchell had made various hypotheses about how transport's supposed to work, and their uniport means that you have a transporter in the membrane that takes a cation, for instance, like lysine or potassium, and allows it to equilibrate with the membrane potential. Has nothing to do with the pH gradient. You can't drive anything with a positive charge with a pH gradient, okay? For acids, we call it symport A, one type of symport, meaning that the proton is on the acid, the protonated acid, as I told you before, only now you have a transporter that transports it in because it gets in much faster with a transporter than it does by passive diffusion. And inside it dissociates and is coupled to delta pH only. You can't, if it was coupled to the membrane potential, it would push the anion out, okay? Symport B means this is like lac permease or anything neutral. The proton goes on the protein, the transporter, goes down the electrochemical gradient, and that free energy is used to drive the uphill transport of a substrate, like lactose, proline, serine, glycine, etc. Antiport means the free energy released from the downhill movement of the proton drives the exit of antibiotics like tetracycline or calcium or sodium, which the cell doesn't want to have too much of inside, okay? These are specific predictions. Some of them work out, some of them don't. The ones that work out are really neat. Now you can see from the data machine what you can do with this. If you took a transport substrate like an acid and you put it at acid pH, you would expect to see stimulation when you add valinomycin under these conditions, right? Inhibition by nigericin. If it's a cation, you'd expect to see the opposite. Inhibition here, stimulation there, etc. and so forth, right? Okay, and this is basically what was done. And I'll show you what it looks like, because it's cool. So you're looking here at again, the data machine. Here's with vancomycin and nigiricin. CCCP kills everything, so we don't have to worry about that. These are systems, this is what you're looking at is data that really was obtained with lactose. But proline, serine, glycine, tyrosine, glutamate, an acid, lysine, a cation, leucine, cysteine, and succinated dicarboxylic acid all look more or less like they're coupled to delta mu H. So vancomycin causes pretty much like that. Nigiricin does this pretty much like that. And when you go to pH 7.5, where the only driving part of the driving force that's there is the membrane potential, vancomycin inhibits like crazy, and nigiricin doesn't do anything. Okay? That's one class of transporters. The other class is really interesting. So here's the data machine again, just so you can look at it. And we move over, and these are acids. So you're looking at data for glucose 6-phosphate, but lactate, glucuronate, and gluconate all do the same thing. Here, when you have the system at pH 5.5, you get stimulation when you add vancomycin, big-time stimulation, indicating that they're coupled primarily to delta pH, like Mitchell would predict. Okay. At pH 7.5, however, there is no delta pH, but they're transported actually better at pH 7.5 than they are at 5.5. And furthermore, vancomycin becomes a dramatic inhibitor instead of stimulating. So clearly, these things are coupled, these acids are coupled to delta pH at 1 pH and to the membrane potential at a higher pH. So you can draw either one of two conclusions from this. Either Peter Mitchell's Nobel Prize was pH dependent, or if you've been burnt once by not believing in Peter Mitchell, you come up with the chemiosmotic explanation for this. Well, the explanation, which is pure hypothesis at this point and will probably remain that way, here's Sophia who did these experiments, is this is the way it might work. So under acidic conditions, the acid would be transported with Z valence and Z protons bound. You'd form a binary complex which would come across, dissociate, and as I told you, the inside being alkaline, you'd cause dissociation of the acid and accumulation of the anion. In order to make this work electrogenically, all you have to do is say there's a functional group in the protein that has to be neutralized that has a pK below 7.5. So here it becomes a symporter. That is, it becomes a symporter A rather, or B rather. So you take the protonated acid plus a proton, and it's that proton that makes it go electrogenically. You make a ternary complex, comes across, the dissociation of the protons and the acid occur, and you go around this way. And this, is a possibility that is really tough to prove, okay? And I don't know if it'll ever be proven or tested, really. But it's an interesting possibility. The stoichiometry has to change somehow for this to work, for it to work the way it works. The other big puzzle that was involved in these days was E. coli does not have a sodium pump. And yet there are transport systems in E. coli and lots of other bacteria that are sodium coupled, not proton coupled. And the way they work is very interesting. So you have a respiratory chain in the membrane that pumps out protons. Here would be D-lactic dehydrogenase pumping out protons. This proton electrochemical gradient is converted into a sodium electrochemical gradient by the action of the sodium proton antiporter. So this is an electrogenic system in which two protons go in for every sodium that comes out, okay? And that converts the electrochemical proton gradient into an electrochemical sodium gradient, which then allows the energy trapped from the downhill movement of sodium with the electrochemical gradient to drive the uphill transport of things like thiomethylgalactoside by MEL-B, as it's called, the protein that does this. And this work was done by Hajime Tokuda, who's now retired professor of biochemistry at Tokyo University. And finally, I have to show you this slide, which is the late 1970s was the end of an era, and this is my lab at that time at the Roche Institute. This is my technician, Lake Patel, Eitana Padan, my secretary, Catherine Tai, Gerard LeBlanc, Greg Kazarowski, Shimon Schuldiner. You don't need to know all the guys. There's me with a cigar in my mouth, and Gilad Rimon there. And that's the end. Thank you very much for listening. I don't think anybody fell asleep. So I hope I kept you all awake. I didn't see anybody dozing, especially Tzipora. So any questions, comments, arguments? What would you like?
So Ron, I was going to ask you a question about the
Alkylophiles, yes. Yeah.
Is there any more insights on how they can
Make high energy phosphate, oxidase?
How they can have a proton-driven?
Well, I think they do the same. They have very effective sodium proton antiporters. That all their transport systems in alkylophiles are sodium dependent. So all they depend on is the membrane potential, right? The pH gradient won't do anything. So you convert the electrochemical proton gradient into electrochemical sodium gradient. The problem with alkylophiles has always been and still is that they have a reverse delta pH, okay? So instead of being alkaline inside, they're acid inside, okay? Because of the action of the sodium proton antiporter. So between the reverse pH gradient and the membrane potential, they only have about 60 millivolts net delta mu H, which is too little to make ATP. Okay, so this has been a big problem, and that a good friend of mine, Terry Krolwich, has been studying for many years. And I guess it's really never been solved. I mean, they think these days, I believe, that this critical glutamate that's in the A subunit is in a very hydrophobic environment. And that's what they think is going on. I mean, it's not clear to me that the problem's solved by any means. So it remains a big bioenergetic problem, how the alkylophiles make ATP. Transport's not a problem, because it's all sodium coupled. The oxphos is not sodium coupled, as it is in some alkylophiles. Like Peter Dimroth discovered sodium dependent F1FO. That exists, but it's not in the alkylophiles that Terry Krolwich studies. That's proton dependent. So you either have to have a stoichiometry of 30 protons per ATP, which is utterly ridiculous, or there's some trick that the thing pulls, which is not clear yet what it is exactly. Yes?
In the kind of experiments you do, let's say taking up lactate, does the total osmotic potential inside the vesicles remain more or less constant, or is there a potential for changing the volume, anything like that during these experiments?
Well, it's a good question. They probably, the volume stays constant. But you have to remember that the respiratory chain pumps out protons much more rapidly than any one of these transport systems, or in fact the combination of transport systems. So in order to see what I showed you, this collapse of the delta pH or the delta psi when transport occurs, you have to really add the ionophore to make sure you only have one component. You won't see it. If you let it go without adding that ionophore, you won't see anything, because because the respiratory chain is pumping out protons so much more rapidly than the transporters are letting the substrate in. Now, the question about volume is an interesting question, which really hasn't been studied in any kind of detail. You might expect that since this stuff is supposed to be in free solution, there's a paper that was published, it must be 50 years ago, in which, I can't remember the guy's name. There was a paper published where he measured by light scattering spheroplasts and looked at lactose uptake and claimed that the spheroplast swelled. Now, this obviously should be done with vesicles, but we've really just never done it. And I don't know, if water goes in, it goes in passively. I don't think it can go through the protein. Okay? Does that answer your question enough?
I'm interested in build up very.
No, that's what I figured. Yeah, they get.
Gain
Well, these may well shrink and swell. They may, but we just have not looked at it. It's something that should be looked at. Yeah, I mean, this stuff is from a long time ago. Okay, tomorrow you'll get the real stuff that I do. Yes, or do now. Yeah, go ahead.
This may not be related, but some of your work reminds me a little bit about, in eukaryotic cells, vesicular transport. As you know, vesicles do have various proton ATPases, etc.
Sure.
And they certainly in autophagy, the acidification of some of these vesicles is very important. And so I'm just wondering, in your mind, does this work have any implications for the biogenesis pathways that autophagy is using in order to transport pro-inflammatory cytokines out of cells? It's considered to be a new mechanism, a new anti-inflammatory mechanism potentially in eukaryotic cells. So I'm just wondering if any of this work in your mind can lead to some insights into the vesicular biology in eukaryotic cells.
Well, now what you're talking about now, fusion events, you know, like synaptosome.
Right.
Yeah, I think it has a great deal to do with the storage and packaging of catecholamines, for instance, for sure. I mean, that's been shown. If you know, had I been smart, really, I would have taken these vesicles. Incidentally, are still the best characterized vesicles that exist. Okay, and had I been smart, I would have moved from vesicles like E. coli vesicles to synaptosomes, for instance. Okay, but I got too obsessed with how transport's really supposed to work on a mechanistic level. But to answer your question, yeah, I think it has a lot to do with what goes on. A great deal. It's just it's the inverse of everything. It's the vacuolar ATPase, synaptosomal ATPase pumps protons into the cell, into the vesicle, and that reverse proton gradient is basically exchanged with catecholamines, for instance, in the chromaffin granules or synaptosomes, and that's how they accumulate. And then they basically complex with ATP to make. They're not really in free solution. They're bound in this ATP amine form. But the fusion event that occurs, I don't know whether that may. I would suspect that the fusion events that take place, for instance, with synaptosomes, probably doesn't work if you collapse the pH gradient, the delta mu H, across the synaptosomal membrane. But that stuff must have been done. I'm sure people used to use a few years ago, before they got all tied up with all the proteins that are involved in the fusion event, things that would collapse pH gradients. And I'm pretty sure that they inhibit release of these neurogenic amines. I'm pretty sure. So I think there's a great deal to do. Once nature discovers how to do something, it's very clear that it conserves it, it keeps it. And that becomes even clearer when you get to the protein level and the mechanistic level. So I would not be surprised at all if these kinds of things, I'm sure they're important to answer your question. The short answer is yes. Yeah.
Ronald, I'd like to go back a long time ago, as you said, things were done, and I'd like to talk about that six-pack, if you will, of power players. Must have been David Green, and must have been Racker, and must have been Munninger.
And Chance, and Lardy.
Chance, and the most exciting part of the Federation meetings in Atlantic City was going and hearing the controversy. Have you ever spoken with any of them about how they felt after Peter Mitchell won the...
Well, I can tell you, you remember R.J.P. Williams?
Yeah.
Okay, so R.J.P., when I came up with my respiratory intermediate hypothesis, called me a hero, okay? And then when I came up with all the stuff really showing that Mitchell was right, and incontrovertibly so, I presented this at a meeting in Oxford, and he walked up to me afterwards and called me a traitor. Okay? And I looked at him like he was nuts, which he probably is. But he claims that Mitchell stole the idea from him. But yeah, they wrote, remember the issue of annual reviews of biochemistry where they all wrote, including Paul Boyer, who I know pretty well. They all wrote chapters, sort of bowing to Mitchell, agreeing that chemiosmosis. It was so clear by that time. For me, it happened in a period of flow dialysis is what did it. I mean, flow dialysis. Here's this. It was just amazing that all this stuff was sitting there. And you just had to get the right method for seeing it. I mean, the membrane potential was fine. That happened first with the radioactive lipophilic ions, and that sort of it was a big step in the right direction. But the stoichiometries would have had to be too high, for it to really be the only thing that occurred. So when it turned out that the pH gradient was there too, when you finally got around to doing the right kind of an assay, then it really became clear. I mean, you can quantitatively demonstrate. I didn't want to take you through all the numbers, but the concentration gradients that you get are entirely commensurate with the amount of millivolts that are there. I mean, if you pretend lac permease is a cation, and you say, okay, how many millivolts of transport do you get? It's easily within the range of the delta mu H, easily. Some of them are really interesting, though. Some of them you get like at relatively alkaline pH at 7.5. I'm like, the stoichiometry must change for some of them. Now, at one point we played around with lactose and thought we had shown that the stoichiometry changed at high pH, because Mitchell, when I sent Mitchell this business with glucose 6-phosphate and those acids, he said, no, this can't be right, because the transport of the weak acid should uncouple, okay? And at that time I bought everything he said, so I said, oh yeah, it may be. So we started measuring this, and it looked like the stoichiometry for lactose went up, which was baloney, okay? And then it occurred to me, look, the respiratory chain pumps out protons much more rapidly than any one of these damn transport systems. So if you increase the stoichiometry for one of them, it wouldn't make any difference at all, because the protons are getting pumped out very, very rapidly. So, that's about where it sits. I think they all gave in eventually.
Did you ever talk with David Green, who seemed to have more vitriol and stuff like that?
Oh God, yes. Unfortunately, I'm glad I spoke to David Green before I really did all this stuff. I mean, it was at a stage when I thought, it was a respiratory intermediate, and he invited me to his lab, and I'll never forget it. It was like a visit to the Mad Hatter. I mean, I sat down, and he stared me in the face, and he said, I've disproven the bilayer theory of membranes. And he said, oh, really? So he said, come with me. So he drags me to his drawing board, and he had all these drawings there, about how the membrane was made up of subunits. You remember those days when he had subunits of the membrane? And it was like, it was a real adventure.
We got one more question over here.
Yes, sure.
So you talked a lot about protons going this way and compounds going that way. It's sort of a black box looking thing where you describe the thermodynamics and it's all beautiful that way. But what about mechanism of action, like in the coupling factor?
You want mechanism? You come tomorrow, and I'll give you mechanism. Okay? I'll give you plenty of mechanism, more than you want. I really almost think I know how the damn thing works, but I could give you, yeah.
I'll come
Tomorrow, okay. You don't want me to start on that now, believe me. Yes. Who?
Dave, did you have?
No,
Anybody else?
that was my signal for one more question.
Well, look. Thank you all for staying awake. How long did I go on for?